All right, welcome all. I'm Dr. Charlie Roberts. I'm Cancer Center Director here at St. Jude Children's Research Hospital, and it's my pleasure to welcome you today to the Science of Childhood Cancer and our latest installment. Just before we begin today's lecture, I mentioned that next week will be Dr. Scott Armstrong from the Dana-Farber Cancer Institute, who will be presenting chromatin complexes as therapeutic opportunities in cancer. So it is a true delight for me to welcome Dr. Richard Gilbertson, at least virtually, back to St. Jude Children's Research Hospital. I would not actually be here at St. Jude were it not for Richard. He was Cancer Center Director here, and it was only after he decided to move to Cambridge University that a search was initiated, and here I am. So I thank him deeply for providing this opportunity to me. But even more importantly, I thank him profusely to be a mentor and a colleague. I've called him up a few times since I arrived to get his input and guidance, and he's been spectacular, always willing to spend time and provide thoughtful and insightful comments. So Dr. Gilbertson holds the Li Ka-shing Chair of Oncology at the department, and head of the Department of Oncology and Director of Cancer Research UK's major center at the University of Cambridge. He did his training at the University of Newcastle upon Tyne, bachelor's there, already working on medulloblastoma, on to MBBS, MRCP, and PhD. He went on to study pediatric oncology at the Royal Victoria Infirmary at Newcastle upon Tyne, and then in 2000 he came to St. Jude, where he rapidly rose through the ranks, becoming a full member or full professor in 2009. He was co-leader of the neurobiology and brain tumor program at St. Jude, and from 2011 to 14 served as Cancer Center Director, and importantly was the first to lead the institution to the rating of exceptional from the National Cancer Institute, the top possible rating, so a real achievement. He also served for Scientific Director as 2 years, holding both positions. He's truly a world leader in studying medulloblastoma and pediatric brain cancers. His work is remarkably impactful, from laboratory studies all the way into clinical trials. He's published many top impact papers. He's elected to American Association of Clinical Investigators, European Academy of Cancer Sciences, Fellow of the Academy of Medical Sciences. He's also one of the most impactful speakers that I've seen, and it's always both a pleasure and intellectually stimulating to hear what he has to say. And so before I turn it over to Dr. Gilbertson, just a quick mention that our series, The Science of Childhood Cancers, now has more than 5,000 registrants representing more than 100 countries all over the world. Thank you all for joining us. And I think it's because we have top quality speakers. And right now I'm going to throw it to one of the best, Richard.
Great, thank you very much, Joe. It's a fantastic introduction. See if I can share my screen. Let me see if that's working. So it's fantastic to be back virtually at St. Jude. I do miss it. It was great working there. And you know, at the risk of being mutually backslapping, I can't think of a better person to have handed the reins of the centre to. And all right, I was the first person to take it to exceptional, but Charlie repeated that with a higher score, I would say. So well done, Charlie. So I'm going to talk to you mainly about unpublished work today. And the reason why I'm using the somewhat bizarre title of Will We Ever Cure Cancer is because what I wanted to do is use the examples of what we learned from children's cancer really to take this into all ages. So if I can get this to work. So I do want us to pause at the beginning and think, what are the big challenges facing us in terms of cancer as a disease across all ages? So we know, this is from the AACR, it's a number of years old now, that the number of cases are increasing significantly, and they will do for the foreseeable future, as was the associated healthcare costs. Tragically, in this country, and I think it's similar in the US, a quarter of all patients with cancer are diagnosed in the ER. All those patients die. That's just simply not good enough. If you look at a country like the UK, we might think we've done very well with children's cancer, improving our insights into it, dividing it into increasingly small subgroups. But that means that patients with some certain cancers, there are only 2 a year in the UK. That's not sustainable for a clinical trials approach. We know the history of drug development. The vast majority of compounds that make it into a druggable form never make it to the clinic. And also something close to my heart, if we look at a country like Africa and other lower middle income countries, if you look at those, you're 5 times more likely to die of cancer if you're a child than if you're born in a developed country. And this is all unworkable and unsustainable. So what are the big opportunities? And I like this graph because it demonstrates life expectancy going back to about 1850 all the way up to current day. And there's this progressive increase in life expectancy. There's a dip here, which is associated with Spanish flu. I think we'll see a similar dip with COVID, maybe not so large, but we'll see that. But there's been this upward trajectory in survival across all age groups. And this has been driven by primary prevention, not by intervention of disease. And I think it's a really big lesson for those of us studying cancer, and that's why I'm going to end my talk, and I'll get there in the end, I promise. Because traditionally we focused on this end of the disease, whether we're working on adult cancer or whether we're working on children's cancer. But we've paid little attention to where cancer actually comes from. And I want to spend the first part of my talk addressing that question. Where does cancer come from in the first place? Now, I'm going to skip over a lot of work that we've done in the past, but for those people who are not familiar with the work that my lab's done, which will be many people, of course, it might serve as a useful introduction. So when I first started working at St. Jude, I owe basically 2 great things to St. Jude. One is it turned me into a decent thinker. I was pretty stupid when I came to St. Jude, but I had some great mentors in the like of Chuck Scher, Tom Curran, many others who really taught me how to think clearly, but also the resources at St. Jude and the freedom to use them really was an exciting opportunity. And those 2 really, I think, launched me on my career. But when I was there, I thought that we were starting to do gene expression profile of kiddies' tumors. And what I realized when I was looking through these lists, that many of these genes actually were normal developmental regulatory genes. So we started to look at the expression profiles in mouse as a model of mammalian development and simply compare using old-fashioned F-metrics arrays what the gene expression profiles in temporally and topographically restricted cells were relative to human tumors in a cross-species approach. And in doing that, we were able to show various things, and one of them was that Wnt medullos, that those who treat brain tumors will know as a highly survivable form of medulloblastoma, doesn't actually arise in the cerebellum, but arises from the dorsal brainstem. And that's contrasted with the hedgehog form. And in human tumors shown in this MRI, you can see this mouse model shown on the left, human tumor on the right, very closely phenocopied and different to the hedgehog subtype. Now, we've got very excited about this, and we've used a sort of cross-species transcriptomic approach to look for the origins of medulloblastoma, ependymoma, and choroplexus carcinoma. But really, all we did was replicate the remarkable work of Percival Bailey and Harvey Cushing, working about 100 years ago with nothing but the aid of a microscope and a few simple stains. And they did a remarkable piece of work by just using morphology. They were able to demonstrate that indeed the roots of these brain tumors in children could be traced back to developmental origins within specific cell types in the nervous system. This is a beautiful review from Paul Northcott, which summarizes a lot of the work that many labs have done around the world. And basically, medulloblastoma can divide it into these 4 subtypes, and you can't spend 2 minutes in St. Jude without hearing this. But really, all you have to know at the moment is kiddies with Wnt tumors, they all survive. Kiddies with group 3, most of those patients sadly die. And hedgehog tumors are somewhere in the middle. And we know that had Harvey Cushing and Percival Bailey had access to gene expression profiling, they'd have done this, and in a much more elegant way than myself, Stephen Pfister, Michael Taylor, Paul Northcott, the rest of the gang who've done this. But what Percival Bailey and Harvey Cushing couldn't have done with that technology is ask the important question, why? So why is it that we have very restricted tumors that have different origins and very different mutations? So why can't a Catenian mutation, for example, so named for the Wnt tumor, why can't that happen in a hedgehog tumor and vice versa? And this is what's beautiful, if there's anything beautiful about children's cancer, because of its closeness to development, I think it can teach really important lessons about how cancer originates and how lineages restrict mutational patterns. And so what we've set out to do is ask that question. How is it that you could have a bunch of cells, and could be any tissue, one of those cells will be highly susceptible to a mutation and it will generate a tumor. But if you put the same mutation in cells nearby, they're somehow resistant to it. And there are beautiful studies done actually from the Sanger in Cambridge looking at eyelids. For those of you who've seen that, you get large clones of KRAS P53 mutant cells, but they're completely normal. They're not transformed. We know that from the gut as well. So what is it that restricts those lineages? And the window into this, and we started this work when I was still at St. Jude, was discovered by Giles Robinson when he was working in my lab, that the second most frequently mutated gene in medulloblastoma is in a gene called DDX3X. And we were interested in that because DDX3X was one mutation that occurred recurrently with high prevalence in two very distinct tumor types. So what we thought was that could tell us something about how lineages are restricted when cancer forms. So what does DDX3X do? This is again from Giles's paper, and he showed that the Wnt mutations, the mutations in DDX, they are at the interface of the connections with nucleic acids, because this is an RNA helicase. And in this paper, distributed throughout the open reading frame. But beautiful work from Cold Spring Harbor has also identified these mutations as germline mutations in the most frequently inherited form of neurological disability and developmental disorders in females. And so here you've got an example here where you've got an MRI scan. We were very excited to see this because of the, basically you can see there's a hindbrain malformation. And of course, when you've got mutation in the gene which affects medulloblastoma, it's always very interesting to see that there is an effect on the hindbrain in humans. So what does DDX3X actually do? So when we were working at St. Jude still, we were able to work with an incredibly smart person you might know. He's called J. Paul Taylor, of course, and a fantastic lab. And we had a really great collaboration with J. Paul's lab. And we were able to show with J. Paul that these mutations actually disrupt or drive super stress granule formation and disrupt the translation of proteins. So it looked like these mutations could have an impact on the translatome, the proteome expression in these cells. But with another St. Jude collaboration, started when we were still there and continued when we came to Cambridge with Tiro. Now, I can't spell inflammasome, but Tiro knows a lot about them. But using our mouse models and some of our expertise on DDX3X, what Tiro was able to show was that DDX3X also has this incredible function where it shuttles between the NLRP3 inflammasome and also stress granules. When it gets incorporated into stress granules, it has a pro-survival response. When it's incorporated into inflammasomes, it's a pro-death response. So there's this remarkable situation where the question is, does DDX3X mutation disrupt gene expression or does it change the stress response during medulloblastoma tumorigenesis? So Deanna Patmore began this work when she was in my lab at St. Jude, and we continued it when we got to Cambridge. And what we do normally in my lab is we'll develop conditional knockouts of genes of interest, and we did that with DDX3X because it's a sex-linked gene, homozygous females that contain the conditional allele complete knockout, heterozygous partial, and males, of course, don't have another allele, so they have a complete knockout. Now, we were very excited to see that when you delete that gene, no other tumor genes, no other oncogenes or tumor suppressors, you simply delete that gene, you get a profound phenotype in these mice. And actually, it models precisely what you see in the human. You get this profound impact on the hindbrain with poor cerebellar development. You get all the lineages present, but there's a high restriction in the number of those cells which actually generate the cerebellum, suggesting there's an early defect in the progenitor population, perhaps an increased cell death. So you're not actually getting the expansion in the cerebellum that's very typical in the first few days in postnatal life. So what might DDX3X be doing in controlling hindbrain specification? So what Deanna went on to do was to isolate hindbrains by microdissecting them throughout development. And what we were surprised to see is very early on in mid-embryogenesis, there was an exquisite loss of just a very small cassette of about 23 genes. But that gene was those genes were the entire Hox cassette that control the rhombomer patterning in the hindbrain. For those of you who remember your embryology, the Hox cassette is very important for laying down the rhombomer patterning, which decide the fate of all of the different progenitors and therefore their progeny, which make up and knit together the hindbrain. Of course, that's really exciting because by deleting the single gene, it suggests, in fact, that what that was critical for was related to Hox transcript expression and the downstream fate of these cells. We've got a good idea about how this happens now, but I don't have time to go into that today. So if it changes the fate of cells, how does it work in the context of tumorigenesis? So while we were at St. Jude, I showed you that model of Wnt medulo that Paul Gibson had made. So we bred our mice that had conditional DDX allele onto mice that had conditional activation of catenin so that they would get Wnt medulos. And we did the same with the hedgehog conditional patch allele. What was very exciting to see is that we did see an increase in penetrance, confirming that indeed it's likely that this is a tumor suppressor gene. But far more surprising was the change in fate of the tumors or the topographical positioning. So I mentioned already that Wnt medulos are highly restricted to the lower rhombic lip. This is an extreme example where these tumors arise in mossy fiber precursor cells, and they actually end up being sort of a cerebellopontine angle tumor with a relatively preserved cerebellum. In the hedgehog setting, we actually see these tumors completely confined to the cerebellum, shown in the bottom left. I actually don't know if you've seen my pointer. But actually, when we inactivate DDX3X, now we get a complete switch. And the Wnt tumors form in the cerebellum, and the hedgehog tumors form in the lower rhombic lip. And you can demonstrate that in the form of heat maps shown here with a large number of animals. So what we were seeing is this fate switch, where in fact, although Wnt tumors would normally be restricted in their lineage to the lower rhombic lip, and hedgehog tumors would be normally restricted in their lineage to the cerebellum, we now saw the capacity with loss of this DDX3X to form tumors in both locations. So what's going on there? And just to give you an insight into that, we basically, thanks Charlie, you can see the pointer there, thanks Charlie, it's helpful. So this is just a bit of gene expression profiling, looking at Wnt signaling, stress response and mitosis from microdissected hindbrains at E16, P5 and P13, and in mice that are wild type, heterozygous for DDX3X, males that have lost DDX3X, or flox females that are carrying this predisposition to Wnt medulloblastoma. Now, what you can see when Wnt is preserved, when DDX3X is preserved, when you activate Wnt signaling, there's this marked stress response, downregulation of mitosis, which explains why we don't get cerebellar tumors forming in the context of an intact DDX3X. But when you lose DDX3X, you no longer see the stress response. Mitosis is allowed to continue as normal. So DDX3X then is lost, and so you don't get this eliciting stress response and tumors form. So the model we have for this is basically, it's in context of wild type DDX3X, which is a stress response signal, probably through that mechanism that Tur has described, shuttling between stress granules in the inflammasome. The activation of Wnt signals in the cerebellum elicits a strong stress response and death, so no tumor generation, so tumor suppression that you can form with hedgehog tumors, vice versa in the lower rhombic lip. And of course, when DDX3X is lost, you can form tumors anywhere because there is no longer the cell stress sensing and a resultant tumor suppressor response. So when I was still at St. Jude, I've always been plagued by a bigger question beyond the brain tumors, and that is, why are cancers distributed unevenly in time and space? I doubt Jim Downey's listening to this, but I used to bang on about this all the time, and I'm sure I drove him mad. But basically, as we develop as humans, and actually it's true in all species that are predisposed to cancer, the onset of malignancies is absolutely prescribed, both in their site and their incidence. So cancer's not about bad luck. It's obviously basically has a blueprint in the context of development. So I'm going to share with you some briefly published data that we've done a while ago when we were at St. Jude, when Lee Chin, who now has her own lab, of course, at St. Jude, was a postdoc in my lab, because it's a backdrop for the main part of my talk, which is all basically unpublished data. So when Lee Chin was in my lab, we wanted to make a model of looking at what was then the sort of big phenomenon in the sort of noughties of cancer stem cells. And that was based on CD133. You know, CD133 was a stem cell marker. You've got tumors express stem cell markers. You could take those out of a human, put them in mice, and only the 133 cells would basically make tumors. Now, we know that's largely rubbish, but what we wanted to do was show that through a completely single species system. So Lee Chin made a prominent 1 mouse in which CRE-RT2, iris, and LAT-Z is knocked into the prominent 1 locus. So basically, wherever the stem cell marker was being expressed, you could actually cause changes in conditional alleles, including lineage tracing markers. This is just from her paper we published in 2009, lineage tracing the small intestinal progenitor population. Now, this fabulous mathematician, who I think is still with you, Arzu Onar-Thomas, was basically the person who invented all the mathematics for this, which was terrific. So this is a published story, so I'm going to flip through it quite quickly. We published it a few years ago. But what Li Qin did was ask the question, which is really the ground state for understanding the relationship between normal development and cancer, is if you want to target a population of cells to understand its normal development, can you then in parallel do the same thing with that same population and understand its cancer risk? Now, we chose the prominent one mouse partly because we made it and we had it, but also what Li Qin had observed was that prominent one in some tissue marked stem cells, in other tissues marked post-mitotic fully differentiated cells. So you could be simultaneously in a single organ activating lineage tracing or oncogenes in a population of stem cells, but perhaps in the organ next door, you could do it in a different, more differentiated population. And then you could compare cancer risk in those different organs. So Li Qin did a very extensive lineage tracing experiment to identify the stem cell function of each of those cells in 12 organs, and I'll talk about that in a minute. But also in a very large study using 10 different tumor suppressors or oncogene conditional alleles that covered the waterfront of human mutations, then asked cancer risk questions. And to sort of cut a long story short, we were able to basically across 12 organs manage to look at basically in neonates and adults, which cells were stem cells using classic lineage tracing, the gold standard, and then looking at the labeling of daughters that had post-mitotic fully differentiated transcriptomes and also proteomes. And then it looked also in those same cells at cancer risk. Now, this is all published, so I'm not going to dwell on it. But the most exciting piece of data we generated from that is this, and this is Arzu Onar-Thomas's beautiful mathematics. And so what this data summarizes is something that's long been suspected, but had never actually been, I think, demonstrated effectively or convincingly in an experimental setting. And that is the notion that juvenile stem cells are intrinsically resistant to cancer. So the green graph here labels as you increase in your lifelong genetic capacity. So what you're looking at here is real data. It's all organs. It's agnostic for organ site. It's basically all data. And it's looking at whether a particular PROM1 population, what its lifelong genetic capacity, you can think about that as a degree of stemness or progenitor, the capacity to generate that organ over the life of the animal. And you can plot that against the lifelong risk of that one cell making cancer. And what was remarkable about this is the result for neonates. So stem cells labeled as neonates, and these are cells labeled in adults. Now, what was interesting is if you take a given amount of stemness, if you like, 25%, then the neonatal cell with that level of stemness across the life of the animal has only about a 10% lifelong risk of making cancer. However, for the same level of stemness, an adult cell actually has a 7 times increased risk of developing cancer. Now, this isn't about a cell having mutations for a very long period of time, because remember, we introduced the mutations in neonates just shortly after birth, and they're still protected against cancer throughout the life of that animal. And the adult cells, which inherit these much later, P60 to 180, depending on when we did the induction, they have a much higher risk of cancer. So this gets at the question I want to really address for the last part of my talk, which is all unpublished data, is could this explain why children don't get more cancer? So one of the things I've been obsessed with ever since I had the misfortune of seeing first patient die of cancer as a child is why is it that children don't get much cancer? So I've shown this graph before at St. Jude. This is supposed to be a kiddie, but this is growth. So basically you're growing more quickly in utero than any other time in life. That growth rate plummets. You have this growth rate in adolescence, and then you essentially don't grow for the rest of your life, apart from you get fat, of course. And this is the cancer risk. Now, the notion here has always been that cancer risk is delayed until you're later in life, simply because it takes that long to accrue enough mutations in the right cell types to make cancer. Now, I think there's obviously some truth in that, but it's largely bollocks. It doesn't make any sense, because if that was the case, solely the case, then cancer would be much more random, and we wouldn't see these patterns that suggest there is some developmental background going on to this. So children obviously have all these insults: mitotic stress, differentiation stress, lots of external virals and other pathogens, and yet they're highly resistant to cancer. And for a long time, there's been this hypothesis that there must be an evolutionary mechanism for multi-organ species to become multi-organ species. In other words, as we were developing as multi-organ species, during that critical phase of early development, when there was a huge burden of mitosis, a huge burden of differentiation, and that organism had to preserve its gametes to breed, to pass on those genes, there must have been a mechanism that would be active just at that period to allow cancer not to form, to be cancer preventative. Now, as you get older, like me and Charlie, that's less important. And so there's no need to have that mechanism in place. So if we understood this, could we get to that thing I said at the beginning, is could we prevent cancer in adults? And for the rest of the talk, I'm going to concentrate on sharing that data with you. And this is the work of a fantastic postdoc in my lab, Amir, who came to work with me in Cambridge. He initially came as a lab technician, but he was too good at the interview, so we made him a postdoc immediately. And one thing that's remarkable about Amir is not only is he fantastic at wet lab science, but he's taught himself completely the bioinformatics and the deep learning approaches that we're using to study this. So what we've done is go back to those same 12 organs, and the brain is appropriately positioned over Amir's head here. So the 12 organs, so we're looking purposefully small intestine, stomach, lung, large intestine, skin, uterus, etc. And there's a really important message, prostate rather, and then kidney. There's a really important message in this, in that I think when one is looking at even cancer origins, or even just looking at cancer itself, if you study a single organ, that's fine, you can get really deep knowledge on that, but there'll be very specific abnormalities or very specific transcriptomic changes, methylomic changes associated with an organ. It's very difficult then to generalize that across a whole organism. So are there rules that govern cancer prevention if such a mechanism exists that are true for all tissues? And of course, it's very unlikely that tissues would invent for each individual tissue a mechanism that would govern that. That doesn't make evolutionary sense. It doesn't make sort of energy efficiency sense. But unless you study those entire organs, you'll not be able to see those commonalities in how organs operate to suppress cancer. Now, of course, there will be organ context relationships to that, but we would imagine there will be sort of underlying principles that one can observe. So the way that we've done this is that we've gone back to the same model, the prominent one model, and the reason we've done that is because of all the work of Li Chin. We've already mapped out what is their lifelong stem cell capacity, what is the lifelong cancer risk. We've done that functionally. We know what those fundamental characteristics are that are associated with these cells. What we don't have is what is the transcriptome associated with that cell, the methylome associated with that cell, the proteome associated with that cell, throughout all stages of normality. But thanks to the work of Amir, we do now have that. So what we've done is for all 12 organs, we've gone in and isolated very early on, immediately after initial induction of lineage tracing, just 3 days later, isolated those cells by FACS. And we've now subjected each of those to proteome, transcriptome, and methylome analysis. So the proteome we've done with Rudy Aebersold, now retired, but fantastic Swiss biochemist who we've just got the proteome analysis back from. Our transcriptomic analysis, we started working with a good friend here, Wolf Wright, who works at the Babraham campus. We're now doing that actually all here in the building, the CI, and we're just starting to do the methylome analysis. I'm going to share with you today the transcriptome analysis, because I think the principles at the base of this will be the same. So looking here at about 1,000 samples from all of these organs, and they're obviously in this UMAP, highly restricted and compressed. So although there are 1,000 here, they're overlapping. Now, they're all basically geographically located or topographically located in this UMAP, based specifically on the organ of origin. You might think, well, that's not surprising. It actually did surprise me, because actually most of these are very small populations of cells and very immature cells within those organs. So even though we're, for example, in the small intestine, just taking this fraction of cells from the crypt, or here in the liver, just taking this fraction of portal cells, which line the less than 0.001% of cells in the liver, or just taking the cells lining the bronchioles in the lung, the UMAP organizes those based on origin. So there is still a fundamental understanding or information in these cells telling us where they come from. Now, the challenge with this sort of data is that not only do we have the transcriptome, but we also, of course, have the proteome, the methylome, we have temporal information, when were these cells taken, we have sex information, we also have knowledge of their cancer risk, and actually, as I'll show you in a minute, we also have done this in damage. And this is a truly big data problem. It's not possible using Excel sheets, if you still use those, or even sort of rudimentary or even fairly sophisticated network or bioinformatic analysis to get at the grips of this data. And so what we've turned to analyze this data is neural networks. So for those of you not familiar with neural networks or deep learning, I'm just going to give you a brief overview of this. So essentially the way neural networks work is they're essentially agnostic approaches to train a sort of the network or the computer system to learn what what defines or what characterizes a particular input. So it could be an age of an organ, it could be an organ type, it could be a cancer risk. So you'll take a group, a set of information, and you'll give those labels at the beginning. And then as the data flows through the neural network, it impinges upon different layers, and you can have any number of layers within there. And they're consisting of these nodes. And these nodes are where bits of information are either prioritized or deprioritized based on whether they're actually giving information about what that original input was. And then it will spit out that information into outputs, and it will be able to essentially train the network. Now, the beautiful thing about this is it is a learning process. So if you add more samples in, if you get more data in, or if you get more labels going into there, it will get better and better at understanding and basically what and recognizing an unlabeled sample and basically being able to bin it into a particular output. So this is just a view of the neural network, for example, for the analysis we've done so far, looking at organs. And I mentioned that these simple cells will have associated features that are indicative and grouping them together based on the organ type they came from. And you can delve into this and you can see, of course, lungs. You see gratifyingly there are markers here like NKX2-1, which is associated with lungs, liver the same, prostate, which is a classic X3.1 Michael demonstrated, and that is associated with the stem cell and prostate. So you can sort of give a sense check that this is right. Now, how does the network work and how good is it at doing things like predicting organ and age? So you can train the network on a set of your samples, and then you can put unknown samples in here and ask, is it any good at predicting what the organ is? And it's extremely good. It's not surprising looking at the UMAP that it's particularly good at predicting organ type. What's more surprising to me is it's extremely good at predicting age. So I can take a liver cell from a P3 or a liver cell from 1 year old or a lung cell, and remember these are organ agnostic, these are all organs, and it will know how old that cell is, even though it's come from 1 of 12 organs, and this is trained purely on age. So what's remarkable is these cells across the body from that small fraction in an organ agnostic way have encoded within them in their transcriptome what age they are. So what about cancer risk? And that's where it gets really exciting. So it actually predicts cancer risk. So we know, because we've already measured, as I've mentioned in Lee Chin's work, we know what the cancer risk is because we've already measured that functionally by exposing these cells to a myriad of different tumor suppressors or oncogenes and measured their lifelong cancer risk. Now, what's remarkable is that you can train the network to predict what cancer risk does, and it does it incredibly crisply. So here are adults, induction, or neonates. The code here represents basically their associated intrinsic stemness and their risk of cancer. Now, neonates have a low risk of cancer, we've already mentioned that. Adults have a high risk of cancer, but there are cells in here that have low stemness or no stemness, and they're no risk of cancer, and actually they predict very well. So this is a fantastic tool, and we're looking at this with companies potentially like Grail and others about whether this could be used to actually detect cancer risk. But I mentioned the issue of damage. So cancer is not just bad luck, and only someone who doesn't think would suggest it's bad luck purely. So going back to our liver model, we demonstrated as before that these rare cancers, these rare stem cells live around the portal system in the liver, and basically they carry some of the markers that are known to be associated with stemness in liver. And we mentioned that in the neonate, these are highly active stem cells. So they exist just around the portal system in the liver in the neonate, and if you just lineage trace them, within 180 days, the entire organ is GFP positive. So this small population of cells basically make the entire liver, remarkably generative, but they become quiescent in adulthood and they don't do anything. However, if you damage the liver, these cells, which we've demonstrated through pulse chasing experiments, are indeed the progeny of those neonatal cells. If you damage them, they actually reactivate and it will repair the entire liver. So this is a population which is highly active as a stem cell population as a neonate, becomes quiescent as an adult, but when the liver gets damaged, they will reactivate and they will actually repair the liver. Now, what's remarkable is if you look at the transcriptome of those, and this is again taken from Lee Chin's paper, actually they're remarkably similar to the neonatal ancestor that made the liver. So this suggests that what happens in an adult is you don't have adult cells that just sit around and they remain as adult cells, but they have the capacity to repair. But they're quiescent, and when you damage the organ, they wake up and they go, hang on a minute, I used to make the liver. So rather than reinvent something new as an adult, there's a retrograde step going back to its neonatal ancestor state, is able to make the liver again. But it doesn't do it very well. It just scars it, but it is able to remake and repair the liver. Now, there are significant chunks of that transcriptome missing. It's not that there are bits of it that don't look like the neonate. So it's not completely perfect in its replicating the neonatal state. Not only that, along with this ability to repair the liver comes this high risk of cancer. So what we're getting at here is that we think in the context of damage, you will wake up these cells, they're there to repair and regenerate an organ, but actually in the context of a mutation, there's also a high risk of cancer, perhaps because they don't have the mechanism to suppress malignancy that existed in the neonate. So that's the liver. This is Lee Chin's data. Is it true for other organs? And again, that's what we're looking at. So the idea is, just to reiterate this, is that you've got stem cells that exist in a neonate. Their job is to make an organ. Now, when they're insulted, they can resist that, and so you can have a low incidence of cancer. Now, in adults, you have similar cells. They've actually, often in our case, as we've seen in the mouse, they're the daughters of the original parent, which was in the neonate, but they've gone to sleep, they're quiescent. Now, in the context of damage, they will wake up and repair the organ, but they're missing, that's the theory, the bit that would actually prevent cancer from happening. So there's also an increased risk of cancer happening. So could we reactivate in the neonate that cassette that prevents cancer from happening, and there you would have a cancer preventative? Now, if that was true, we should actually be able to show that this is generalized across organs, going back to that point. I've shown it in the liver, but the liver's a bit funky. Ask Lee Chin, because she still studies it. The liver's got, last count, 20 or 30 candidate stem cells in it. But what about organs like the lung or organs like the large bowel? So that's exactly what we've gone on to do. So I'm going to share that with you now. So understanding cell fate shifts and damage. So we've repeated the work in the liver, but we've also now done it in the lung and the large bowel. So I mentioned early on, you've got this lineages that come from stem cells, but about P60 in the mouse, all these cell populations essentially go to sleep, they become quiescent. Now, what we're doing is damaging those with a tissue-specific damage that's appropriate, DSS in the large bowel, and then naphthalene in the lung, and then chasing those with our neural networks to ask what happens to the cell state when we've damaged that. Do we see an increased risk in cancer predisposition? Do we see a change in age? I'm just going to share that data with you now. So again, to remind you, the system is extremely good. The neural network is extremely good at predicting age. And this is just data from damaged lung and large bowel, which was damaged between P60 and 180. Now, what's remarkable is that, yes, a significant chunk of the transcriptome, the characteristic of these cells, are still identified as being P60. But also a large component of this is basically to the left. In other words, there's a retrograde becoming younger in terms of the transcriptome, suggesting these cells are adapting that earlier state that we saw in the liver. So what about cancer risk? So this is another way of looking at the data, and this is basically a measure of belonging to a particular class. Each of these individual points is a sample, and this is relative to the prediction of being in a class. So obviously, these are predicted to be in this class here, which is basically quiescent and non-cancer risk. And this is the class which is a high cancer risk, intrinsically high cancer risk. So in an undamaged lung and liver, there's a high prediction that these samples, these cells taken from these undamaged large bowels and lung, are of a low cancer risk. There's a very low prediction that they'll be high cancer risk. Now, if we take the same cells out of a damaged organ, we see this indecision. There's this remarkable system where the neural network says, yes, okay, they still have a sort of mixed characteristic associated with this lower risk, but now there's a much higher prediction that they're associated with actually high cancer risk. So just in the context of damage, there are no oncogenes or tumor suppressors in this set. By simply reactivating those cells, not only have we regressed their chronological age, if you like, down to an earlier state, but also we've increased the risk enshrined within their transcriptomes. So it's delving into this data that we think is really going to help us to understand what are those core transcriptomes that ultimately across multiple organs define what is cancer risk, and we can discuss why that might be helpful both for intervention strategies but also for diagnostic strategies. Okay, that's all very well, but how do we avoid gene lists? This is a photograph of the first Affymetrix array set that I got from St. Jude. It was really exciting when you got St. Jude in 2000, so 21 years ago, which I can't believe. We were doing our first expression array experiments, and you would ring up people and say, is the data ready? Is the data ready? And then eventually the data would arrive on a card, and it was this. And it was just lists and lists and lists of genes, all on dot matrix printer. And of course, you would rank those genes, and you could transfer them into obviously Excel sheets and other. But really, that's pretty pedestrian, because actually, for this same ranking of genes here, they actually don't look like a league table, like a sort of sports league table, top team to bottom team. The relationship is actually more like this. So how do we use our neural network data to not just devolve this into a list of genes, but actually into a living feature in which we can start to truly understand the interrelationships, not just between genes, but between age and cancer risk. So we don't want to just turn our neural network into this list. And that's where we're using topographical and temporal relationships using Euclidean distance. So I'll just give you an example of that. If you just take 1,300 genes and you ask the system to organize these genes based on their relative correlation to one another at a particular state. Now, this is actually labeled by age in terms of color, that doesn't matter. And it's the output, it's the correlations between genes, and the tightness indicates this is particular a group that defines a particular characteristic within a given aspect of that cell. Now, what's cool is when we haven't told the system anything, but if you look at this purple class, it's all the cell cycle genes. So this neural network is self-organizing the biology into something that's real. Now, most of these other features are not known in terms of their biology. So we're using a completely agnostic approach with this deep learning system to get the biology to be self-organized, to tell us what the biology is, rather than us going in and hierarchically in supervised way and saying, this is a list of genes. Now, if you do that for everything, it gets really pretty. So this is looking at all of those samples that Amir has taken and now organizing what we're calling the sort of universe of aging. And it shows over time how there is a difference and a relationship, and how these relationships between genes change as an organ ages. And you can layer onto this information related to cancer risk. So you can really start to see how these interactions between transcriptomic changes happen. Now, we haven't got the proteome in here or the methylome, but you can do the same. So you can you can have the same label for a gene, but add a postfix where it's proteome, transcriptome, and methylome, and then start to understand, for example, how does a transcriptomic change in, say, one set of genes affect the proteome expression of another? Now, how does that work and why is that useful? So I'm just going to use one or two examples to how that might be helpful. So I haven't chosen CD74 for any particular reason, but it gives you an illustration about how these things change over time. So CD74 is one of these, and at P3, the neural network is telling us it correlates with 3 other genes. That goes up to 4 genes at P15, and then it drops off a landscape between P30 and P60. But it becomes really important when this cell differentiates and goes on to P180 a year and 2 years. So that's one gene. If you do this whole transcriptome, it's really cool, and you can start to see these correlations. Now, the trouble with that is that to some extent, it's dependent upon the individual labels that we have for given genes. So one thing that we're using now is something called natural language processing, or learned node semantics. I think this is a really helpful analogy to understand that. So I think you recognize this guy. I think he's being tried at the moment, actually. But anyway, back in 2000, if I said Donald Trump, you might say taxes, scandal, tower, money, stock market. But if I said Donald Trump in 2020, you'd say president, election, Republican, fake news, sex, scandal, tax, all those kind of things. Now, back in 2000, you'd never say president or election or fake news even. So that just shows how over time, this individual gene, not genius, this individual gene has a whole load of semantic language elements associated with it that completely changes with time. And so we're actually using that same approach to interrogate our networks. And actually what's really cool, what Amir's doing, and I'll just remind you that Amir's self-taught all of this. I haven't done any of this, it's Amir, he's done a fantastic job. So you can create these Autobots, so virtual beings that will walk through your network, obviously does it very quickly, and then it associates those and it looks for these semantic and language connectivities between these nodes. And when you look at that, it flattens out sort of into a tree of life, if you like, showing the critical connectivities that happen over time. So what we're hoping, and this is me finished now, so what's the big idea? Well, our big idea is we might not be able to cure cancer, but could we actually get to the point where we prevent it? So I think I've shown you this analogy before, but what we're hoping for is that if we can use these neural network interrogation approaches, I think, first of all, we might be able to identify those transcriptomic, methylomic, or proteomic patterns which actually indicate very high risk for cancer in an organ agnostic way. It's not a huge step to imagine including that in, say, the GRAIL methylome signature or another signature within a blood test. So could you actually identify these signatures 5-6 years beforehand in an individual who's basically incubating an epithelial tumor? But also, if we could understand that, and we're starting to do this with, we hope, a company here and the functional genomics facility, is could we identify what is that cassette that's missing that is present in the neonate that suppresses cancer, but doesn't get reactivated in an adult, so you just repair the tissue. Could we possibly reactivate that pharmacologically? And if we could do, would that be a statin for cancer, a preventative? So I'll pause there. I've probably said enough. Thanks to all the members of my lab as I've gone along, but also the people who funded, including St. Jude. With no doubt, without them, I wouldn't be here today. So thank you very much. And if there are any questions, Charlie, I'd be happy to take them.
Fantastic talk again, Richard. Thank you very much. And just a reminder to attendees, any questions, select all panelists and put them in. So I want to begin with a question, Richard, that strikes me as surprising that the childhood or infant or young cells have sort of a less cancer potential than the adult. It might be the inverse of what I might suspect as, all of this potential, all of this growth opportunity. And so I'm wondering, could that be an intrinsic wiring for the pediatric cells to want to differentiate? There's all of these highly proliferative cells, but in short order, they're going to be differentiating to large parts of the body. And we think about, the classic example of neuroblastoma that can just stop growing and differentiate. So could it really be that, that of almost getting ready for their future that's really distinguishing that compared to adult sort of stem cells that are often just maintenance and will always continue to need that repopulating hematopoietic stem cells, etc., going on?
So I mean, it's a great question. And in fact, in part answer to that, we know actually when you reactivate a quiescent adult cell, it doesn't differentiate off, you don't repair, I mean, you do in the liver to a degree, but most tissues you'll get scarring, right? You won't get a completely beautiful reformed organ. So that's certainly possible. We're actually completely agnostic to the mechanism. It could be non-cell intrinsic, it could be cell extrinsic, it could be immune mediated. And so one of the beauties of looking at the transcriptome and proteome is that you can infer cell-cell communications depending on which aspects are, if you like, not activated. We're also assuming that it's a passive mechanism. It could be that in adults, something is activated for another reason, which makes you pro-cancer. So rather than being something that's not present, so a tumor suppressor mechanism, it could be an inadvertent process of inflammation which is being activated, which predisposes you to being more likely to develop a malignancy. So absolutely. I mean, the short answer is yes. And at the moment, we're completely agnostic to what that would be. I think what's exciting is that the data that Li Chin generated and now Amir's generated is we really feel it's a robust system to start to interrogate that on a really truly large scale.
Great. A question came in. Thinking about, you've talked about preventing cancer with Big Idea. Any thoughts on whether this could have a role in sort of stage 4 or aggressive cancers and treatment options as we think about therapeutic intervention?
Yeah, so the work I've not had a chance to talk about today was there's another study in the lab which came out sort of partly related to this, where we've identified a mechanism we think is responsible for invasion, which is completely distinct from the malignant process itself. So one of the surprises of this sort of scale of data is how it unmasks biology you didn't previously appreciate. And actually that relates to membrane potential. So, which is completely different. I mean, it's a cell intrinsic mechanism, but it's one you would never bet on. And it seems to control sort of the epithelial mesenchymal transition point. And so what we do think is actually those will be important in children who are developing invasive disease. Because again, we have a fairly reductionist approach that might be simplistic, but our bias is that cancers don't invent anything new. They use existing processes, but in a very aberrant way. So I don't think there's going to be mechanisms that are distinct that a child uses in their cancers for invasion relative to an adult cancer. So that's what we'll be looking at for sure.
Great. Question comes in from Giles Robinson. The hypothesis supports cancer being more common as you get older, but it doesn't explain why childhood cancers are not just less common, they're also different than adult cancers. Any thoughts as to why these, how you address this concept?
Yeah, no, I think it's a good question, Giles, even from a Spurs supporter. But I think that the short answer to that, of course, is the pedestrian answers, the context are different. So most children's cancer we know are developing in embryonic tissue. But it does get back to the issue that Charlie raised, which is a good one, is that the differentiation potential of a neonatal or juvenile cell is very different from that of an adult. So because most stem progenitor cells in adults don't have the capacity to regenerate the organ, then it's not surprising perhaps that they're not out in that juvenile state that would also lead on to, say, a neuroblastoma or a rhabdomyosarcoma or an osteosarcoma, because those cells intrinsically, they're in a state which is different. They're there to be quiescent, but just to repair. So they don't have that retained capacity. And it might be the lack of that capacity that Charlie referred to earlier on that could be why they have a higher risk of developing malignancy.
Great. Fabian Luzzi asks, do you know or have some sense of how large the damage needs to be to wake up stem cells? Is there a threshold that's needed to make them arise to trigger this?
Yeah, that's a great question. We don't know that because obviously what we've done is major damage. I mean, I know Amir's done some work on terms of DSS or in terms of DDC or naphthalene dosing to look at the level of damage. Of course, the challenge for us is that because we're taking out organs and then sorting what's already a rare fraction, then you want to have a significant amount of damage so that you're actually capturing those cells, otherwise there aren't enough to analyze. I would say one observation that's interesting is that even in animals that aren't damaged, we see a proportion of animals which are showing this retrograde sort of transcriptome and also perhaps edging towards being increased cancer risk. And my interpretation of that is this is a background damage that's going on in these animals. We're not damaging them, but we're all damaged all the time to a degree. The tissues are undergoing constant damage and repair, and perhaps that's reflected in the level of imperfection that exists in organs.
Great. Any insight on whether gender differences affect the neural network based predictions that you see? Do you see representation of gender, and does it affect the predictions?
Yeah, that's a great question. So gender is certainly included, and that's why I included it in that slide. We do actually see some differences across gender, and what we're hoping to some extent is that might help provide insights into why, as we know, for example, in children, boys tend to have a higher incidence and do worse than girls historically, and that's also true in some adult cancers as well. So we do see some differences, and that's one thing we'll be looking into.
Great. I'm going to ask Dr. Susie Baker to join us as well. She's a co-director of the Neurobiology Brain Tumor Program at St. Jude and Associate Director for Basic Research in the Cancer Center. She's going to help field questions as we go past the hour as well. The next question comes from Yumgu Han, who says, if juvenile stem cells are more resistant than adult stem cells for oncogenesis, why do pediatric cancers have less mutations than adult cancers?
That's a great question. So short answer is we don't know, but we do think that the fundamental mechanisms by which they arise, and this gets back to Charlie's point, which is a great point about the context of oncogenesis happening in different differentiation contexts. So what we don't understand is why some tumors happen in children at all. So if there's a mechanism, so why does it fail? So that's a separate question. So obviously in those children, the mechanism is failing. And one of the suggestions that's come in discussions I've had with a really great developmental biologist here, so we've got a very large stem cell group here, is actually conjoined with an idea that they've had, and that explains why methylation changes are particularly important in children. So the failure that's happening, we think, in some children is a fundamental defect in methylation, which of course could have an impact on differentiation. So we think it's that complex interplay, and that's why we're really excited about including the methylome in the neural network, because the transcriptome has given us all this data, but of course we already have some of the proteome data. I haven't got time to share that with you. But that's different again from the transcriptome. There are some elements that are common, but not that, and then the methylome will be different again. So we're hoping that that complete picture will sort of shed light on that kind of question.
Great. Dr. Gilbertson will continue to answer questions. I want to mention we've reached the official end, but he's delighted to stay along and answer any additional questions, and Dr. Baker will take the lead. Spectacular presentation again, Richard.
Great to see you.
Richard, fantastic presentation, very thought-provoking as always. I'm kind of interested in what might happen if you were to induce damage that causes chronic inflammation rather than sort of an acute damage, since there's a very clear association between inflammatory diseases and like Crohn's disease, for example, or Barrett's esophagus and cancer.
That's a great question. I mean, of course, DSS does that to a degree. It causes sort of severe inflammation in the large bowel, and so we've done it in a sort of very acute sort of onslaught, so you get severe inflammation. But one question we've discussed is, what if we did it at a low level and produced chronic inflammation? Would that pattern be different? So that would be something we'd be interested in looking at, particularly at whether the qualitative changes in the transcriptome are different. And of course, with ulcerative colitis, what's interesting, what's different from Crohn's disease, is that ulcerative colitis tends to be it doesn't have the skip lesions of Crohn's disease. It affects the entire large bowel, whereas Crohn's disease is skip lesions. You get large areas of normality, and of course the cancer risk is much higher in ulcerative colitis. So the question is also, is it a statistics game? It's also simply the number of cells that are being subject to this waking up from quiescence, and of course there's a field effect on the number of mutations. So I think again that gets back to the issue of why having the sort of global approach with the sort of neural network deep learning approach is you can introduce that information. You just simply can't with other approaches.
Excellent. So lots of questions are rolling in here. So one is, have you investigated if there's a shared transcriptional factor network upregulated upon damage between different organ types?
Yes, that's great. So We just got the data yesterday, so actually Amir only generated those graphs for me yesterday. So we are looking at, that's precisely what we want to do, is to see, you know, what is it that's activated across different organs. And again, to emphasize, that's why we've used different organs. So, you know, large bowel, lung, liver, and then of course comparing that to all other organs. It gives us this sort of organ agnostic approach where you actually lose the noise which is organ specific and just retain those bits which are basically common laws across all tissues. So that's what we're hoping to look for.
Are you considering the roles of non-coding RNAs in your models?
Definitely not. Next question. No. So we haven't. It's a great question. Of course, we're not set up in our system to actually sequence for non-coding RNAs. I don't think they are captured by some of the transcriptomics. What I didn't mention is we are looking at single cell as well as bulk. So this is bulk transcriptomics, albeit on a purified population. We are looking at single cells. But it's a great question. We're not looking at that, no.
Have you compared if you get the same network connections with the proteome data or the methylome data and the transcriptome data, or are there some different networks that are developing when you look at
Yeah, so that's a great question. So there are two parts to that. One is I think the short answer is you probably almost certainly will, because we already know, we haven't done the network analysis on the proteome yet, but we do know, as you'd expect, because we know from proteome and transcriptome, of course, they don't correlate. There are some elements of the proteome which are highly expressed along with the transcriptome, and you get inverse relationships all over the place. I think what we're wanting to do is, rather than looking at them separately, is I keep using the word agnostic, but it's useful. Can we just forget what sort of macromolecule it is? It's just simply label them and look at, well, so how does a particular methylation state relate to a particular proteome state? And so, and that's again the beauty of this approach, is you can holistically include, you know, really what are the sort of macromolecular features that underpin these cell types.
Okay. How would you envision cancer prevention in terms of therapeutics? Currently, we discover potential drug targets that drive the cancer in some way. What kind of potential drug targets to prevent cancers may emerge from these studies? Or do you think prevention is something different?
So that's a great question. So there are two, I mean, people who know about prevention and I don't talk about this. So one mechanism is more like a statin. So if we were to identify, you know, let's dream for a moment, there is a pharmacological agent which you could deliver, which tickled up by 10 or 20% the ability of a quiescent cell to reactivate that cassette that it used to when it was a neonate that suppressed cancer. Let's imagine it exists. So you can imagine high populations, you know, so those with familial cancer risk. What if we were to dose those individuals after the age of 50 or 30 with a statin that basically increased that element of the transcriptome? So that's one aspect. What we're also very interested in is sort of the long-term plan for cancer for the NHS here, which I was involved in writing, has a very big ambition that by 2023 we'll have increased the number of survivors by 55,000 by decreasing stage at which cancer's diagnosed. So you can imagine if we find a methylation cassette which really says, look, this person has cells circulating in their blood that indicate a high risk of cancer, you could intensely screen those individuals, particularly if you knew what cell type or organ that was coming from. And therefore, could we detect more cancers earlier? So it's not primary prevention, it's more secondary prevention. So you're intervening at an early stage. So I think that's what we're looking at in terms of the scope of the approach.
Good. That question was from Spencer Curley, I should
have Spencer.
Mentioned, yes.
Yeah, excellent.
Okay,
best looking postdoc I ever had.
One from Anupathania. Do you think that checkpoint mechanisms for controlling proliferation are more prominent or efficient in kids compared to adults, and that's why they have less chance of getting cancer?
That's another great question, and it's certainly a possibility. And we are hoping that we will uncover those sorts of elements within the proteome and transcriptome. And of course, one of the reasons, actually, it's a great question, one of the reasons I showed that homing in on the mitotic element is that we are hoping not only will we see in a supervised approach, because obviously whenever we look at genomics at the moment, how I used to look at genomics, I'm always looking at it through the eyes of sort of supervision, something that existent knowledge and imposing that existing knowledge on the data. The beauty of this approach is that you will see that, but also unknowns. And so will it bring into cell cycle checkpoint, perhaps, you know, some aspect, would it link it somehow to the immune response that you would not expect. And so those aspects we'll be definitely looking at.
Okay, a question from Robert Mobley. With regards to invasiveness in EMT and building a neural network, would trophoblast stem cells or trophectoderm possibly be an important tissue to include? Could trophoblast stem cells, the first cell type to activate EMT in development, hold some important information that we may overlook if we only check tissues of the organism?
Yeah, that's a great question. And of course, there are rare tumors, as Robert will know, that involve the trophoblast. So, you know, tragic tumors that happen in early pregnancy. So we know that. The reason we didn't look at that is simply because we were basing our data on what we should have done. So we knew this was a population based on prominent 1. What we don't know is whether prominent 1 labels those cells which generate the trophoblast. It doesn't mean we can't go in and isolate trophoblast cells per se, or indeed any other stem cell. For example, people will notice that the blood is conspicuous by its absence, and that's because prominent 1 doesn't label the blood, at least not at the stem or progenitor level, so we don't use that in our system. So that's another system. Actually, Scott, who's going to be talking next week or your next session, so he and I are going to be doing that with the blood.
Okay, Ashutosh Mishra says, great talk. Have you had a chance to apply the neural network to brain damage, for example, in Alzheimer's or neurological disorders?
That's a great question. So, so we haven't, but we have a grant through CIK, the New Roads grant, in which we're working with a guy called Rick Livesey, who's a really well-known Alzheimer's researcher based in London. And we're looking basically at generating similar data from developing neonates in humans. And he's got a large bank of single cell data from patients with Alzheimer's. So he's actually interested in doing that. So we haven't done it yet, but he is interested in using the network for that.
Okay, this is a philosophical question, I would say, from Sean Gorby. Looking at recent events, what differences will happen in expectations for a cure for cancer now that we've seen the speed of research for COVID vaccinations? Will this, could this be accelerated with global motivation and funds to make it happen? Could it happen with cancer? What lessons may apply?
That's a great question. Actually, it sounds very name dropping, but I was on a call this morning with Downing Street with the Prime Minister about this to talk about exactly that question. So the question is, you know, he's very excited at the moment because he's seen the response the UK's had to COVID. And so he sees, fortunately for us who are doing research in cancer being the next one that can be solved. I think there's 2 elements to this. There's no question that technologies can be rapidly accelerated, even detection technologies for early cancer, which we're very interested here in Cambridge. What you can't change is the disease. So for example, if you were developing a preventative, that will take 20 to 30 years because you'll have to have that length of time to understand whether this is actually preventing cancer. And so that's why, you know, high risk populations are particularly useful. But in terms, so the short answer is I think it will be very good for accelerating ideas, knowledge development, consolidating that, and actually generating the technologies. But the disease will determine how quickly we roll it out. But I would say, just to Sean's question, is that we did some economic modeling here in the Cancer Centre. So Cambridge invents stuff all the time and we spin it off in companies. But we've worked out that the average delay in spinning out companies and developing new technologies, when you don't have a congregation of those individuals together, is about 5 years. And that's just the mean. So there's a 5-year delay in not congregating people together. So I think what it is going to do is produce an appetite for generating hospitals here where you have physicists, engineers, chemists, mathematicians working alongside, you know, the biomedical and also the clinicians to rapidly accelerate that early phase.
Interesting. Richard, what are the kinetics of the responses that you see? So you're inducing damage. Are you taking them all of your organ stem cells out at a particular time point after damage? And how long does the stem-like susceptible state persist after damage?
That's a great question. I don't know the answer to the second one, but it's a brilliant question. So we basically do it very acutely, so 3 days after damage and tamoxifen induction. So we really are just taking it that acute state when enough time to have an impact of the damage, but not so long. And the reason for that, of course, is as the repair happens, we're using lineage tracing to isolate the cells. You'll be taking the daughters of those initial cells as well, and so it'll get messy. But actually, it's a great question in terms of how long that state persists. I don't know the answer to that, but it is something that we could easily measure. And the other thing that we want to do, although it's very difficult with the animal situation over here, is actually measure cancer risk in the lung once we damage it. Cancer risk in the intestine when we damage it. Of course, we know that for the liver, but we don't know that for the lung and the large bowel.
Yeah, so the neural networks are incredibly powerful and very appealing. Obviously, the entire physiology of cancer development is going to go beyond your Excel spreadsheet list in a one-dimensional priority order. But how do you view validation studies for these kinds of predictions? How can you really how many genes will you have to manipulate or perturb to sort of validate the signatures that you are seeing?
Yeah, that's an excellent question. I mean, I think a simple answer to that is obviously you can repeat the experiments to a degree on a smaller scale and simply repeat the analysis. Is that robust? Now, to some extent, that's built into the fact that we have now 1,000 individual samples. Then also we've got single cell completely separately generated, so you can look for those transcriptomics at the single cell level, and so that in itself gives you a robust sense that this is a valid approach. However, I think what's going to be more complicated, and this is why I didn't get a chance to talk about this, but I'm really excited about it, permutations of, the other thing is permutations in neural networks. So if you see critical nodes, can you actually do experiments in silico where you would perturb a gene and see what happens to the network as it ages? So can you actually, and then do it multiple times, multiple permutations, and then look for stability in those changes. And so that will then be much more directed at going in and testing that in a directed way. Because obviously what we'd like to do is find critical genes or nodes, which I don't believe will exist because the system would be ridiculously fragile. Yes. But are there nodes that we can actually go in and perturb? But I think a first step will be these multiple permutations of an in silico modeling, and I think they will be really useful, I think.
And you are identifying, I assume, the sort of stem cell signatures that your earlier work had showed was associated with cancer. So what's the proportion of kind of new players that are together in the nodes with those previously?
Yeah, I don't know the answer to that. So we certainly are seeing previously unexpected connections. So that's another great thing about it. It's actually Abcam is quite a famous antibody company that are on our campus. And they span out from Cambridge. They're interested in talking to us because, of course, they want to make antibodies. So if we do identify, particularly in the proteome, exciting new markers, then we'll make lots of money.
Good for everyone.
Yeah, absolutely.
There's a question from Emily Seiden. Why have you specifically chosen to focus on the methylome? Are there other important epigenomic changes like between pediatric and adult cancer?
Yeah, that's a really good question. So the reason for choosing the methylome is because I don't know very much about epigenetics. No, it's because we can measure it at the scale we're looking at, essentially. It doesn't mean we couldn't go in a directed way with a sort of chip approach and look for other aspects. But look certainly at the scale, because it lends itself to sequencing. So we can do bisulfite sequencing on very small amounts of tissue, which is why Wolf's particularly helpful as an expert at that. So we can actually go in and do that. And we're actually looking at doing it potentially at the single cell level as well. So you can do that kind of scale sequencing.
I think we have hit a nice wide spectrum of different topics with the questions from the audience, which was I just looked at it at one stage was over 480 people.
That's more than I ever got when I talked at St. Jude.
So, Richard, this was really a wonderful seminar, beautiful work, and very thought-provoking, obviously, due to the terrific range of different kinds of questions. So it's been a delight to have you back at St. Jude, if only on the screen.
It's lovely to be here. I really enjoyed it. Thank you.
Yes, thank you so much for joining us today, and thanks to all of the people in the audience. Just a reminder again, the the next seminar next week will be from Scott Armstrong, chromatin complexes as therapeutic opportunities in cancer. So thank you so much, Richard.
Cheers. Nice to see you all. Take care.
In person when the air clears. Yeah.
Absolutely. Brilliant. Take care. Nice to see you all.